All right, so this is podcast 23, part B. We have been talking about the flow of a nerve impulse from the dendritic or dendrite end of the neuron towards the end of the axon. Okay? Let me get rolling here. Sorry. There we go. All right. We had said that it's like there are many protein channels embedded in the cell membrane of a neuron. They are ion channels in that they are allowing ions to pass. The ions that are passing are sodium and potassium. The sodium ion channels we need to think of as being like dominoes, okay? So the sodium ion channels are like dominoes, okay? And once we stimulate one enough to open, let's say, for example, we stimulate it to open right here, okay? The normal polarity of the membrane, let's say we've got this set up as normal polarity. So let me scribble these out for a second, okay? And we stimulate a channel here, sodium channel to open. Then the sodium positive ions will flow into the neuron. And they will flow in at such a high amount that we will alter this to be negative, and now this will be positive because so many sodium ions came in. This change in polarity will cause this channel to open right here, and sodium will flow in here. And then that change in polarity will cause this sodium channel to open, and a change in polarity will now cause this one to open, and that one to open, and that one to open, and we will flop the polarity all the way down the neuron. Okay? Stimulated by this, potassium channels that also are embedded in the neuron membrane will also open to attempt to restore this original charge difference back to more positives on the outside and negatives on the inside by allowing positive potassium ions, positive ions, to flow out, thus making it return to being more negative inside. Now, we do this so that the nerve impulse does go in the right direction towards the end of the axon and not backwards back up the dendrite. Okay? Now, oh, we already did this. Okay, there we go. Okay. I think we've already gone over all of that. Okay, that's a good slide, though. It's a nice synopsis, so pause this if you need a synopsis and get that information. Okay. Now, this is sort of set up back with the polarity that we need. You can see that we're following this change in polarity, and it's going to change all the way down, but we're resetting it. But you should notice there's something that's not exactly like it was set in the first place. Okay? We'll talk about that in just a second. But I do want you to know the name of this region where the flip-flop of the charges is moving down the axon. This region is called the action potential, okay? So this is where we are depolarizing this neuron, if you will, okay? It is called the action potential. Something is happening here, okay? And it's propagating. That means it's moving down the axon, okay? This action potential can move from your brain down to your fingertips in an incredibly fast rate of speed, milliseconds. Now, how miraculous is that? It is absolutely astounding to think of that happening so fast, such that we can think and talk and do all the things that we can do. Okay, so action potential, very important term for you to know. These ion channels that we have been talking about are called voltage-gated channels because this alteration in the voltage, I should be pointing right here, is what causes the next one to open. It's what stimulates it, so they're called voltage-gated channels. They open as a response to voltage changing around them. Okay? And here's a slide that says, but Houston, we have a problem. This is what I was trying to allude to just a few moments ago. We are trying to reset this neuron so that we have to reset this neuron so that we're back to having positives on the outside and negatives on the inside. And the opening of the potassium channels that we talked about right behind the opening of the sodium channels, it does that. It gets these positives out so that we restore the positive being on the outside and the negative being on the inside. But that's not exactly how we started. We started with lots of positive potassiums on the inside and lots of positive sodiums on the outside. And we're flip-flopping that. Even though our positive negative is back in the right place, our sodiums and potassiums are flip-flopped. And so this neuron cannot fire until that is fixed. Okay? The sodiums and potassiums are on the wrong sides. We were able to prevent the charge from going backwards by doing that, but we still can't fire this neuron again until we fix them and put the sodiums and potassiums on the right sides. So we need to move sodium back out where it needs to be and pull the potassium back in. Now, in order to do that, we are going to have to move them against their concentration gradients. Okay? Even though we let sodium rush in, okay, there's still more sodium out here than there is inside this neuron. So in order for it to come back out, we can open the door all day long, but they're not going to flow out because there's still more out here than there is in there. That's just the chemistry of the surrounding tissue. Okay? So we've got to move these things against their concentration gradients. And in order to do that, we need a pump. We need a protein pump that will push the sodium and the potassium against their concentration gradients. Okay? And there is a pump that exists to do just this thing. And it's called a sodium-potassium pump. Okay? The sodium-potassium pump is active transport. So if you are asked for an example of active transport, here it is, very vital in the neuron. It's not the only place where there are sodium-potassium pumps, but clearly it's a really important place because there's a whole lot of movement of sodium and potassium here to allow that electrical impulse to flow down the neuron. Okay? As a pump, as active transport, it absolutely requires ATP and lots of it. Okay? For every ATP that is spent, every energy molecule that is spent to move this pump, 3 sodiums are pushed out and 2 potassiums are pushed in against their concentration gradient. Okay? And over time, as the pump works, we're going to reset the proper sodium and potassium gradient inside and outside of the cell. Okay? And it takes a lot of energy. So this is one of the reasons you eat, is so you will have energy so that these pumps can work and keep your neurons firing the way they should. Okay? Now, once the pumps act and push the potassium back in and push the sodium back out, then that neuron is set up and ready to fire again. Okay? We've got everything back the way it should be, including we've got the potassium in and the sodium out. like it was supposed to be. Okay? This is called resting potential. This neuron is at rest and ready to fire again if we stimulate a sodium ion channel to open. If we knock over that first domino, it will do the whole thing again. Now, as we are moving, here's your neuron again, and as we start out, we're starting out positive on the outside, negative on the inside. We tickle a sodium gated channel and it opens, and so we are going to reverse that charge. Okay, so this amount of charge reversal and change as we go down the neuron can be measured. Okay, and that's what this graph is showing here. Okay, at number 1 we are seeing would be time. I don't know why they didn't label the axis, but this is time. So as we start the nerve impulse, we are at resting potential. The nerve's not doing anything, and we already said that's a negative 70 millivolts. So that's a lot of difference in charge from the outside to the inside of the membrane, relatively speaking. Okay, now if a stimulus reaches threshold potential, okay, then we are going to depolarize. Okay, so we're hitting threshold potential. We are going to depolarize that neuron at that point where the stimulus was received. The sodium channels will open, okay, and allow sodium to rush in. And you can see as sodium comes in, it is getting less and less negative inside. And indeed, not only does it get depolarized, meaning we have no charge change between the inside and the outside, but in fact, it goes kind of overboard. We get a lot of sodiums on the inside, enough to actually cause there to be a plus 30 charge difference between the inside and the outside. So we kind of let in so much sodium that we kind of go overboard a little bit in our depolarization. Okay? Now, then on the other side of this hill, as the potassium channels open, we begin to repolarize the membrane. So we're pushing out, or actually allowing to flow out these potassium ions, and so we're losing positive charge on the inside, so we're bringing it back down to the point where we're at negative 70, and in fact we let so many potassiums out that we actually overshoot our original negative 70 resting potential and actually go down to negative 80. That is called hyperpolarization. We've kind of overdone it. We've allowed so much potassium out, we kind of overshoot the target of negative 70. However, as our sodium-potassium pumps work and so on, we are going to get our normal resting potential back. Okay? So that's what that graph is showing. Okay. Now we need to talk a little bit about this myelin sheath and something it allows to happen. Okay, the axon is covered, as we already mentioned, with cells called Schwann cells. They are made out of myelin, which is a type of insulating fatty substance that allows the axon to be protected and not connect with other neighboring axons and have the charges short out each other, so to speak. But it also does something else. It speeds the signal along. Okay. I want you to picture walking in, I don't know if you can see this or not, I guess I'll find out when I play it, but if you are walking in little baby steps, here I go, I'm baby stepping across the room, okay? Baby stepping. It's going to take me a while to make it from one side of the room to the next, whereas if I leap like that, I'm going to go a lot faster, okay? I'm skipping over huge chunks of floor when I leap across, okay? Well, this myelin sheath, these Schwann cells that are next to each other, allow the neuron to skip, the impulse to skip large chunks of the axon. In between each Schwann cell, and I don't know if you can see with this picture, it's not the best for showing it, but in between each of these little rice grain type things, so to speak, a little bit of the axon is exposed. Okay, you can see a little bit of the axon. These places are called nodes, okay, where the axon is exposed. Where the axon is covered with Schwann cell, the impulse doesn't go through that. It actually skips from here to node, to node, to node, to node, to node, to node, to node. And so instead of that impulse having to travel every little section of that neuron like the baby steps across the floor, instead it can hop to hop to hop to hop to hop. And that makes the impulse travel much more quickly down the neuron. Okay, so this is called saltatory conduction. Saltatory simply means to jump, jumping conduction. We're jumping over large sections of the axon with this. By hopping over, rather than having to touch every single little bit of the axon, we accelerate that nerve impulse. Okay? Indeed, you can see here actual numbers, 150 meters per second, rather than what seems kind of fast to me, but not near as fast as that, 5 meters per second. So 330 miles per hour versus 11 miles per hour. That's a pretty huge difference, okay? That's a much better picture, so you can see actually the nodes there to there, okay? And this is the Schwann cells are being shown here, these layers of fat, okay? In multiple sclerosis disorder in which people lose muscle function and so on, your immune system is attacking the myelin sheath cells, and so you end up with a loss of signal. The wires get crossed, so to speak, and the saltatory conduction is limited, and so your signals move slower. There's confusion in the signals because neurons are next to each other and not insulated. So that's part of the problem with multiple sclerosis. Okay? Now, we have talked at great length about how an impulse would get, say, you feel a stimulus, you put your hand on a hot burner on your oven or something, and so you feel the stimulus enter the dendrite, and it is moving down the neuron through the axon, and it gets to the end of the axon. Now, you want to get this information that you have your hand on a hot burner to your muscles so that you can move your hand off that hot burner, okay? Well, so far we've only gotten it to the end of this neuron. We haven't gotten it anywhere that's going to do us any good. We haven't gotten it to another neuron that might communicate something, or we haven't gotten it to a muscle cell that might actually move our hand out of the way. So we have to deal now with what happens when we reach the end of a neuron, and now we've hit this space, this synapse. How are we going to get the message across the synapse to a cell where maybe that message can do us some good? Okay? This is a great little analogy. How are we going to get, if our abdominos are not close enough to each other that they're going to push each other over, how do we get that message to this domino that he needs to fall to? Okay? We have got to jump that synapse. We have got to get the impulse to go from one cell over to another one. This is a detailed slide. Follow it as best you can. This is really critical to know. Okay? All right. So the action potential, that depolarization event, moves, here's your axon right here, and here is the end of the axon, okay? That action potential moves along the axon, and gets to the end. This space here between this and that That space right there, that's the synapse, that's the space, okay? Now, when the nerve impulse reaches the end of the axon, of course, it is going to depolarize down here just as it depolarized all along on the way, okay? Now, however, at the end of the axon, this depolarization is going to cause something to happen. In the end of the axon, there are calcium channels, and you can see one right there. These aren't anywhere else in the neuron. They're right here at the end of the axon. And the depolarization, the impulse, will cause these calcium channels to open. Now, there is more calcium. Don't ask me why. It just is a fact that there's more calcium out here in the synapse than there is in the neuron. So guess what diffusion will do? There's a high concentration of calcium, a low concentration of calcium. We open the channel. Calcium flows in with its concentration gradient. Okay? Now, what the calcium ions do is they cause little vesicles that are always floating in the end of the axon, these little vesicles, to march up to the end of the axon and fuse with the end of the axon. So this little bubble would come up here and fuse with the end of the axon. Now notice these little vesicles are containing something, these little red dots. These little red dots are neurotransmitters. It is chemicals. And these are actually the guys that are going to carry the chemical, the message of the nerve over to the next cell. Okay? Neurotransmitter, a really, really important word. Neurotransmitter. It's a chemical that is actually going to cross a synapse. Okay? So when the calcium ions stimulate these vesicles, to fuse with the end of the axon. And when they do so, the neurotransmitter that they contain is just released into the synapse. And guess what? It's in a high concentration here, low over here initially, so they flow across the synapse, diffusion with their concentration gradient. Okay? Really important. They diffuse across the synapse with their concentration gradient. Now, once they get to the other side of the synapse, there are little receptors over here that are designed specifically to receive whatever particular neurotransmitter this is. And they will land in these receptors and cause things to happen. Perhaps they will cause another type of ion channel to open on the other side or something like that. Okay? Basically what we've done here is we had an electrical signal zooming down to the end of the axon, and now we converted that electrical signal to a chemical signal, and that chemical signal is leaving here, coming over here, docking and causing something now to happen over here. Perhaps a new channel opens, and so we get a new event happening on this other cell. If you look here, this looks like it's a muscle cell. So maybe we're getting a message from our brain to our muscle to move our stupid hand off the burner. And it is jumping over by way of this neurotransmitter onto our muscle cell and telling us to move our hand. Okay? All right. Now, once we have opened the channel, done whatever we needed to do, then we've got to make sure we take these neurotransmitters out, or else this channel is just going to stay open forever, and that means we can never do this event again. So we've got to clean this out of these little receptors so that we can fire this again and make this muscle move again. Okay? So that's very important, and we're going to talk about that a little bit more as well. Okay? So once we jump that synapse, then the next cell might be another neuron, in which case we would cause a sodium channel to open, and that electrical signal would propagate right on down the neuron. Okay, and we just keep the message going. Or it might be something called an effector, which just means something that does something, like a muscle cell. So the postsynaptic, or after the synapse cell, can be another neuron. It could be a muscle cell we're telling to do something like that. Okay? And this is all just what we said with that drawing. The neurotransmitter, the thing that crossed the synapse, is received by and opens some ion gated channel and causes something to happen in that next cell down the line. Okay? This is just rehashing everything we just talked about. Okay? So here we go again. Now, there are some very well-known neurotransmitters. Please remember, we're talking about things that are leaving from the axon, crossing the synapse, and being received on the other side by a receptor and causing something to happen, perhaps a muscle contraction or whatever, over in a next cell. Acetylcholine is a very famous neurotransmitter, and you should know it. It is the one that stimulates, that jumps from a neuron over to a muscle cell and tells the muscle cell to contract. Really, really important one. If you know no others, you should know that one. Okay? Epinephrine is another neurotransmitter. You might have heard it more familiarly as adrenaline. It causes the fight or flight response. So if your cells receive that, they're going to do something a little bit different. Dopamine is another neurotransmitter. It's widely found in the brain and can affect all kinds of things, as you can see there on the screen. Serotonin is another neurotransmitter, also widespread in the brain and also affecting many things that dopamine affects as well. So if you're interested in that, you can Google those topics and find out all kinds of stuff, I'm sure. All right? But the bottom line that you need to take away from here is that different neurotransmitters are involved in doing different things. The receiving cell, if this is my neuron and this is the cell and in between them is the synapse, there could be certain instances where dopamine is secreted to the next cell, but there are other instances when serotonin is secreted. And there are different receptors over here to receive those different neurotransmitters and thus give this cell a different type of response depending on what gets secreted. So there's a lot of complexity in what can happen between two cells depending on what type of neurotransmitter is sent over and what type of receptors for neurotransmitters exist on that cell that is going to be catching them. Now, neurotransmitters have been discussed as kind of a weak point in the nervous system, in that if there is a substance that affects the neurotransmitter or is maybe similar to it, it can impact nerve function. Let's think about this. I'm going to skip all this. These are things that can mimic or affect neurotransmitters, and they're all things that can do strange things to your brain. Let's look at this. Acetylcholinesterase. Good Lord, that's a big word, but really, it's not that big a deal. All right, acetylcholine is a neurotransmitter that jumps from a neuron to a muscle cell. Okay? ACE is telling us this is an enzyme. Acetylcholinesterase is the enzyme that cleans acetylcholine out of the receptor in which it has landed. So if this is my muscle cell, this is my axon, I sent acetylcholine over, acetylcholine lands on that receptor, and I have to clean it out of the receptor if I ever want this receptor to do its job again, to be able to receive more acetylcholine someday. Okay? So, let's say that this big blob is what acetylcholinesterase looks like. So this big blob would come over here and grab acetylcholine out of that receptor, and then this receptor would be brand new and be able to do whatever it needed to do again. But snakes, for example, and there's lots of things that can do this, can produce something called a neurotoxin that is able to bind into the active site of this enzyme. And this is showing you snake toxin binding into that active site. Well, if something is bound into the active site of that enzyme, then it can't do its job. It can't grab acetylcholine out of that receptor because its active site's already full. So that means once this is fired, once this muscle cell has received that neurotransmitter, it can never fire again, ever, because that acetylcholine is not going to be taken out because acetylcholinesterase cannot do its job because its active site is all full with neurotoxin. That is a classic way in which nerve poisons work, nerve gases used in warfare and all of that kind of stuff, or snake venom. They work by disabling acetylcholinesterase, and thus you end up paralyzed, and all your muscles are paralyzed, including your diaphragm, which means you can't breathe, which means you die. Okay? So neurotoxins inhibit acetylcholinesterase, meaning to inhibit is to prevent something from doing something. Okay? Sarin is a classic neurotoxin used in warfare, horrible thing. All right. So here's some questions to think about. And I would talk with you about these, but this has taken so long that I think I'll just shut up and maybe we can address some of these in class. All right? I hope you have a great rest of your evening or whatever it is as you're watching this. Thanks for watching, and I'll catch you in class. See you later.